Hello students, welcome back. Let us continue where we left off last. Proteins Part 2. When last we met, we stopped with the different types of proteins. There are two basic types of proteins. We have structural proteins, which we call fibrous proteins, and we gave some examples of those, but we did not have time to write those down. And so if you all would, give me two examples of structural proteins or fibrous proteins. Actin, very good. Actin, myosin. Now let's see, can anyone name one more? Myosin. Has anyone ever heard of collagen? Collagen is a fibrous protein, so collagen. And so these are fibrous proteins. They're shaped like ropes. They don't really have, they basically just have almost alpha helical structure only. Nothing else. They aren't folded into a ball like your globular proteins. Now let's look at our next type of protein. Collagen. Collagen is a protein that's found in connective tissue. It's like a long strand like protein, and it's found. It gives your muscles strength. It gives your connective tissue strength. It's also found in bones. You have these things. Think of a bone. Like if I drew a bone here, this is surprising to a lot of people. Like if you have a bone here. Now within a bone, there are like little straws that run the length of the bone. They're like straws inside of straws that run the entire length of the bone. And so if we look at a bone, one of those straws from the top, it would look like this. And so we have these nested straws that run the length of a bone that gives bones compaction strength. Now one thing that's surprising, like if I drew the side of one of these straws here, there are collagen fibers that actually wrap around these straws here. They wrap all the way around these collagen fibers, and they impart strength to your bone, and they prevent your bones from breaking. So collagen is just a very strong protein that gives strength to tissues. Okay. Let's go to the next type of protein. This is fibrous. What's the second type of protein? Functional proteins, we can call them, or globular proteins. Okay, let's see here. Do I have that? Yes. This one here, the one we just did, these are either, we can call these fibrous or structural proteins. I'm going to put this up here with this. Okay. And now let us look at the next type of proteins. Globular proteins. They're called globular proteins because they make a round glob. And so they have their round shape. And so globular proteins. Globular proteins are round. And so if we have a polypeptide here, so if we just had a polypeptide, it would just look like this unfolded. But if we were to fold this polypeptide, it would fold and turn, and then you would have your alpha helices here, and then it would fold some more in your beta pleated sheets. Beta pleated sheet region, fold more. Then you have alpha helices, fold more here. At some points, you can have these proteins where you'll have one cysteine and another cysteine will join together to make a disulfide bond to lock that protein together. Then it'll just fold more. Keep going. Okay, and it'll keep going here, and then that will be your protein. And so this would be the amino end of the protein, and this would be the carboxyl end. So here N and C. And so you can see that this protein has roughly a round shape. Can you see that? It's rounded. And now, for these globular proteins, usually for a globular protein, the outside or the surface of that protein is going to be polar. And so that surface of the protein can have like positive charges. It can have like lysines here and aspartic acids, maybe a histidine here, maybe an arginine, a glutamic acid, a lysine. Maybe it has a what is this? Which one has an OH on it? Maybe it has a serine here with an OH that makes it polar. And maybe it has here another lysine. So the outside of that protein is going to have polar amino acids, whereas the inside of that protein is going to have nonpolar or hydrophobic amino acids. And so that makes this, by putting the polar amino acids on the outside, it makes this protein soluble in water. Does that make sense? Okay. Okay. The outside of the protein, the outside of a protein, a globular protein, because it's in a cell in water, basically, the outside surface needs to have polar amino acids, like lysine or glutamic acid or aspartic acid or arginine or serine or threonine. These amino acids are hydrophilic or polar, so they're going to be on the outside. But on the inside of the on the inside of the protein, where there's no water, basically, you can have your hydrophobic amino acids on the inside. And so by doing this, this protein becomes soluble in water. You want your protein to be soluble in water. Does that make sense? Excellent. Now, sometimes in a protein, can you see, I'm just going to highlight this area here, this yellow, this like little groove here that we actually made here. See that opening there? A lot of times what the proteins will do, they'll when after it folds, a cleft will be formed. And that cleft is going to be, for example, if this is an enzyme, that cleft is going to be an active site. And that's where a substrate will come in. It'll convert that substrate into a product. And so this cleft here on this protein is called an active site cleft, or an active site. So that protein folds in such a way so that it'll make a groove for grabbing the substrate, if it's an enzyme. Does that rationale make sense? Excellent. And so this is a globular protein. Okay. And so globular proteins are like round. Fibrous proteins are long like ropes. Globular proteins are round like balls. Okay. Everyone okay with this one? Let's look at one other type of protein. Okay. I'm going to draw a cell here. Okay. This is going to be a cell. This is the membrane. And then here you have, within the cell, you're going to have its nucleus with its DNA. And then you're going to have a lot of organelles out here. And so this is a cell. Now what I'm going to do, I'm going to blow up, not like explode, blow up, but make larger. I'm going to make larger just a region of the membrane. And so here we're going to have, let's see here if I can draw this well. Okay. Okay. I'm going to draw just a region of the membrane here. A membrane is a bilayer. What are these structures I'm drawing here? Phospholipids, exactly. And so, and let's say this membrane just goes on forever. And so this is just a section of the membrane showing some of the phospholipids. Everyone okay with that? Now I'm going to put I'm going to put a protein in this membrane. Okay, now I'm going to draw a protein here. Okay, I'm just going to draw a protein that would be found in the membrane. And here's a protein that's going to be found in the membrane. Okay. Now, you all should be able to figure out, let's say that the top is going to be the outside of the cell, and the bottom is going to be the inside of the cell. What liquid is found on the inside of a cell? What is that liquid called? Cytoplasm. The cytoplasm, things in the cytoplasm are dissolved in what substance? The particles or the substances in the cytoplasm are dissolved in what liquid? What liquid makes up the cytoplasm? Water. Exactly. So there's water inside of the cell. Okay. And what liquid is found on the outside of cells in the extracellular space? Water. Okay. I know these are trick questions. Okay. So there's water inside of the cell, water on the outside of the cell. Now, is water hydrophobic or hydrophilic? Hydrophilic. Okay, so the polar head, the heads on these phospholipids on the outside of a cell, do they need to be polar or non-polar? Polar. They need to be polar because they need to dissolve the water. So we're going to put here, these need to be polar. All of this needs to be polar because that's where water is. These need to be polar. Okay. What about the phospholipid heads on the inside of the cell? Okay, I'm going to repeat this. What about the phospholipid heads on the inside of the cell? Do these need to be polar or non-polar? I thought I was going to trick you. Exactly. It's going to be polar. Very good. Very good. So it's got to be polar here as well. Okay. Now, the fatty acid tails. Let's make these longer here. The fatty acid tails on phospholipids. Are they polar or non-polar? They're non-polar, exactly. So these are going to be non-polar. And so we'll put here, let's get a good color here. We'll put an N here for non-polar. All of this is non-polar. Okay. Now, here's the million-dollar question. I'm going to shade this here. I'm going to shade this part of the protein yellow, and I'll shade the bottom part down here. I'm going to shade it blue, and we'll leave the middle of the protein white. Now the question is, let's start at the top. The yellow portion of this protein, is it in contact with water, or is the yellow part in contact with the phospholipid tails? It's in contact with water. So this yellow region of the protein, of this membrane protein, does it need to be polar or non-polar? It needs to be polar. So what types of amino acids are going to be found here? What type of amino acids are going to be found in this yellow region? Okay, what's the general name? Okay, lysine is one of them. What's the general name for these types of protein? These types of amino? The hydrophilic amino acids, or the polar amino acids. And so we're going to have things like lysine, which has a positive charge. We're going to have lysine. Let's get a good color, purple. Lysine is going to have a positive charge. Arginine is going to have a positive charge. Glutamic acid negative. Aspartic acid negative. Histidine, depending on the pH, can be positive. And so you're going to have your polar amino acids at the top, or sticking on the outside of the cell. Make sense? Okay. Now, let's look at the blue region. The blue region at the bottom here. This part goes to the inside of the cell. So is this blue region, is it in contact with polar substances, or is it in contact with hydrophobic substances? Polar substances in contact with water, exactly. So what type of amino acids are we going to need to have here? Hydrophilic. Give me a hydrophilic amino acid. Name one. What? Give me a hydrophilic amino acid. Not glycine. Oh, you said lyse. Okay, lysine. Exactly. Exactly. And so you're gonna have polar amino acids here. Make sense? Now let's look at the white region of this transmembrane protein. The white region. Now this white region, is it in contact with polar substances or non-polar substances? Non-polar substances. So what type of amino acids need to be here? Hydrophobic amino acids. Name a hydrophobic amino acid. Glycine, exactly. So these are going to have no charges. These are going to have no charges. Okay, and so this is a membrane protein. So that's how membrane proteins are made. That's the structure of a membrane protein. So membrane proteins are said to be amphipathic. Amphipathic. They have a polar region and a non-polar region. So here we have a membrane protein. And we call them, we can say they're amphipathic. The word amphi means both. Do you know of a word that has the phrase, has the root word amphi in it that denotes two things or both? Ambidextrous is one. It's not spelled exact, but ambidextrous is one. Amphibian, exactly. So these are amphiphilic. They are both polar, and they love water and they love non-water things, organic things. Okay, now I'm going to ask you all a question, and let's see where your thinking is. Name for me a membrane protein. Okay, don't answer it yet. I want you 3 seconds. Name for me a membrane protein. You have 3 seconds to think about it. 1, 2, 3. If you know of one, if you know of a membrane protein, raise your hand. Let's see who has come up with one. Okay, 1, 2, 3 people have come up with a membrane protein. Name it. No, lysine is an amino acid. I want a whole protein. I want a whole protein. Okay? Yes. No, phospholipid is not a protein. The phospholipid is a lipid. I want a protein that's found in the membrane. Calcium ATPase. Exactly, because it lets the calcium in. Name another one. Okay, sodium potassium pump. It's in the membrane. Name another one. We talked about this for 13 lectures. No, not protein kinase A. We have the channels. If I draw it, you're all going to feel like I'm going to draw it anyway. What is that? The adrenergic receptors. All of those adrenergic receptors. We drew all of that time. All of those adrenergic receptors we drew. So the adrenergic receptors, they're membrane proteins. They're in the membrane. And so they have a polar region, which is the part that the catecholamine binds to, and they have a non-polar region where that thing goes up and down the 7 times, and they have an internal hydrophilic region. Now, what binds to the external hydrophilic region of the adrenergic receptor? What binds to the external hydrophilic region? The ligand. What is the ligand? Norepinephrine, okay. What binds to the internal hydrophilic region? The G protein. Now it's starting to make sense? Excellent. Okay, and so that's all we're going to say about membrane proteins. We talked about 3 types of proteins. Fibrous proteins, which are completely structural. Globular proteins, which are rounded. And then membrane proteins, which are amphipathic or amphiphilic. Everyone okay with those? Excellent. Let's talk about levels of structure, levels of protein structure. So we're going to get rid of this one. maybe another. Levels of protein structure. So there are 4 levels of protein structure. If you just have amino acid 1, amino acid 2, amino acid 3, amino acid 4, ... amino acid N. If you just have the sequence of amino acids, just the sequence, what level of protein structure is this? Primary, exactly. This is primary. So this is just the primary protein structure. It only gives you the amino acid sequence. And so an example would be, let's put glycine. Somebody give me another amino acid. We're just going to name any 5 amino acids. Give me 4 more. Histidine, that's a good one. Lysine. Anyone? 2 more amino acids? What was that? Tyrosine. ... And 1 more. Glutamine. Did you say glutamine or glutamate? Glutamine, GLN, glutamine. And so that's a primary amino acid structure. It tells you nothing about how the amino acid is folded or how many polypeptides there are. It's just the amino acid sequence. That's the primary structure. It's sometimes written as 1 superscript O, primary. Everyone okay with this? The second level of protein structure, but before I go to it, I'm going to just represent the primary structure like this. Okay, that's primary structure, just a long strand. Okay, now let's go to secondary structure. Secondary structure, there are 2 secondary structures that you'll need to know. And you all already know what they are. What are the 2 secondary structures? I did not hear you. Alpha helix and beta pleated sheet. Exactly. And so, if you have a protein that just has alpha helix here, if you just have the alpha helix, if only the alpha helix is formed, we call this secondary structure. So this is alpha helix. So you can have either alpha helix or you can have beta pleated sheets. And so with beta pleated sheets, what happens? The protein goes along and then it just goes like this. It turns back on itself. And it makes like a sheet actually, which makes sense. And so this is going to be beta pleated sheet. And so these 2 make secondary structure. So if a polypeptide only has beta pleated sheets and alpha helices, only secondary structure is formed. Only secondary. It's not completely folded yet. And so we can write secondary like this: 2 naught or 2 prime. And that's secondary structure. And so here, if you have primary structure, you just have a polypeptide. This is still just a polypeptide. It's not a protein. It's not a protein. Here, secondary structure. If we just have secondary structure, do we have a polypeptide or a protein? It's still a polypeptide. Okay. Let's go on to the next structure here. If you have a protein where you have your alpha helices and beta pleated sheets and additional folding, and And this protein, if this is a fold, now this polypeptide is folded. A folded polypeptide. Now we can give this another name. A folded polypeptide, we can call it something else now. What can we call it? A functional protein. Sometimes we can call it a functional protein. We'll put a question mark here. Sometimes. And so, what type of structure is this called? Tertiary. This is tertiary structure, and it can be written 3-0. So we have a folded polypeptide. Most of the time, we call a folded polypeptide a functional protein. Now, I don't want to have to draw all of this every time here, because that's going to be kind of complex to try to draw every time. So if you all would allow me, I'm gonna say that all of this equals how should I draw it? Equals this. And so this is my folded polypeptide now. Is that okay with everyone if I do that? It's just going to make it easier. Okay? And so that's tertiary structure. The protein is completely folded. Now we can go to our last, bless you, we can go to our last level of structure. Sometimes, if you just have one polypeptide, one folded polypeptide, the protein is still not functional. Sometimes you have to put several of these together. And so if you have this has to join. You actually have to put several of them together here. And now you have 4 folded proteins together. Did everyone follow me on that? Because I did do a lot. Okay. And so here we have 4 folded proteins. So we have 4 folded polypeptides. And this type of structure here, where you have 4 folded polypeptides, it doesn't have to be 4. It could be, actually, let me change something because I don't want to confuse you. This might confuse you. Let's do something. If you all would allow me, if you all would be so kind, I'm going to remove one. Well, did anybody write with ink? Okay, let's do this then. Let's do this then. Instead of removing one, let's just give it 5. Okay, and so this is not 4 folded. That's going to be 5 folded, because I don't want you to confuse something. So we're going to have 5 folded proteins, and those five folded polypeptides make one functional protein. It makes one functional protein, and we call such a structure. Anybody have any idea? Quaternary. And so this is going to be quaternary. Q-U-A-T-E-R. Quaternary. N-A-R-Y. And that's 4-0. And I didn't want to put 4 quaternary with 4 folded polypeptides, because you would think it's only quaternary if there are 4 of them. That's why I put a 5th one. Does that make sense? Because I don't want you to get confused. And so the 5th level of structure is called quaternary. Now, if a protein has more than one polypeptide, we call such a protein a multimer. A multimer. M-U-L-T-I-M-E-R. Or more than one unit. Has more than one unit. if a protein has more than one folded polypeptide, like here we have 5, 1, 2, 3, 4, 5, we call that protein a multimer, many-mer units. It has many units, a multimer. Make sense? A polymer is going to be like an amino acid. When you put a lot of amino acids together, an amino acid is a single entity, but a protein is a polypeptide is a larger structure made. It is a polymer itself. And so if you think about it this way, if you put a lot of individual units together like an amino acid, you make a polymer, like a polypeptide is a polymer. If you put many of those polymers together, now you have a multimer. I mean, it's really semantic. It's really semantic, and this can confuse students a lot. And so here we have a multimer. Sometimes all the units of that multimer are the same. Sometimes they're different. And so I'm going to show you I'm just going to use this as our example here. So let's make ourselves a multimer of just 2 polypeptides. Okay, so here 1 and 2. So here we have a protein with 2 polypeptides. Is this a multimer? Yes, it's a multimer. And so it has two polypeptides, and so we can call this multimer a dimer. A dimer, because it's 2 units. 2 units. Di means 2, mer means unit. And so this is a dimer. Make sense? Now I'm going to draw something else. So let's see if you all can figure out what this one is. What is this one? Is this a multimer? Okay, this is a multimer. What type of multimer? A trimer, exactly. Okay, I'm going to do one more here. 1, 2, 3. Okay, is this a multimer? What is it? What type of multimer? A tetramer. A tetramer, exactly. Could it be simpler? Can I do anything to make this simpler? No, it's not possible. It's not possible. Excellent. Now, one last thing. So this is all part of quaternary structure because we have more than one polypeptide together. Now I'm going to do something else. Okay. This polypeptide here, this first one here in this dimer, it's going to be red, and the second one in the dimer is going to be green. So the 2 units, even though they're still joined together, they're different. They're different. Okay, and let's just do another one up here. Okay, I'm going to draw another one. I should have just copied that one, but I wasn't thinking clearly. So we're going to put 2 here that are the same. And so both of these are dimer. A is a dimer and B is a dimer. A, the dimers are the same. The units are the same. B, the units are different. They're still dimers. Everyone okay with that? So now we have to come up with a name to distinguish between a dimer where both units are the same and a dimer where the 2 units are different. Everyone with me? Can anyone help me to come up with such a name? Okay, for this one then, you're saying we can call this one the alpha subunit. We can call this one the beta since they're different. And that's what they do. That's actually what they do with what scientists do with proteins. They're always called the first protein the alpha subunit, and the second one, if it's different, they call it the beta. But if they're both the same, we call this one alpha. What would we call this one here? Alpha. So they're both alpha. But we still need to come up with a name to distinguish between those 2 types of proteins. Exactly. And so we're going to call this first one here a homodimer, because both of them are the same. a homodimer, H-O-M-O dimer. And we'll call the second one here B. What type of dimer? A heterodimer. Can it be simpler? No, it can't be. It can't be simpler than this. And so this is quaternary structure. And so as we have this trimer, as we have it written here, is this what type of trimer would this be? It's a homotrimer. But you really don't get into proteins that are homotrimeric or tetratrimeric, or tetra or homotetrameric. Usually at that point, when they start getting that many subunits, they're going to be different. Okay, everyone okay with this? And so that's quaternary structure. And those are the 4 levels of protein structure. Everyone okay? Questions? Okay, put your notes away. I don't know. This didn't work last time. I'm going to see what happens again. Okay. As far as to go. Oh, there it goes. Wonderful. Okay. Let's see here. What is the percentage of protein in a cell? Ooh, that's unfair, Dr. Merritt. A dehydrated cell. More than 50%. Very good, very good. What is the name of the process by which you take messenger RNA and you make a polypeptide? Translation. Very good, very good. What is the name of round proteins that like enzymes? What general name? Globular proteins. Name a membrane protein. Any membrane protein. Adrenergic receptor. Very good. Membrane proteins have a hydrophilic region and they have a hydrophobic region. For this reason, they're given such proteins are given a special name, or such substances are given a special name. Amphiphilic or amphipathic. Very good, very good. Beta pleated sheets and alpha helices. There are 4 levels of protein structure. Which one are these? Secondary. Let's say you have a protein. It has an alpha subunit and it has a beta subunit. What level of protein structure is this? Quaternary. Quaternary is a level of protein structure. What type of protein is this if you have an alpha unit and a beta? Heterodimer. Have a great day, you all.